Hello everyone. Today we are going to discuss about gram staining technique. So already we have discussed about simple staining technique, a very simple technique. And today we are going to discuss about gram staining, which is also called as differential staining technique. So let's see what is there in the PPT. So gram staining comes under differential staining technique, and this differential staining is a type of staining in which more than one staining is used. So this type of staining is very important to bacteria. Then what is the importance of doing gram staining? In simple staining, we can able to observe the morphology and arrangement of the bacteria. But in gram staining, as it names telling like it is a differential staining technique, we can able to distinguish the bacteria into gram positive and gram negative bacteria. And coming under differential staining, we can also see acid fast staining or Gill-Nielsen technique. So this acid fast staining is also one of the example for differential staining technique, where we can able to distinguish the bacteria into acid fast bacteria and non-acid fast bacteria. So like this, gram staining will be helpful to know us whether our bacteria is gram positive or gram negative. So I'll explain what is gram positive nature, gram negative nature in the coming slides. So this procedure divide the bacteria into, as just now I explained you, like with the help of gram stain reaction we can able to distinguish them. And this is differential staining procedure because it divides into gram positive and gram negative groups, the most widely employed staining method in bacteriology, because this is the basic thing where we can able to distinguish the bacteria whether it is gram positive bacteria or gram negative bacteria. Very important method to distinguish. So gram positive bacteria will stain purple or violet color, whereas gram negative bacteria stains pink or red in color. And the scientist who developed this gram staining technique is Hans Christian Gram in 1884, and he belongs to Danish, and he is a bacteriologist. Then coming to the experiment process, so our aim is to perform gram staining technique and to differentiate the bacteria into gram positive and gram negative. So what are the requirements we need? We need clean glass slide, we need bacterial culture, we need crystal violet and saffronin, and we need grams iodine, we need 75% ethanol, we need distilled water, and also microscope. So if you see here, we are using two stains, that is crystal violet and saffronin. So crystal violet is nothing but the primary stain, saffronin is secondary stain, whereas grams iodine is also called as mordant, and 75% ethanol is a decolorizing agent. So with the help of this we are going to perform the gram staining technique. So coming to the principle behind gram staining, generally bacteria stains differently because of the fundamental differences in the structure and composition of their cell wall. So if you see gram positive bacteria, their cell wall consists of thick peptidoglycan layer and also teichoic acids in the middle. Because of the presence of this thick peptidoglycan layer, which is made up of N-acetyl glucosamine and N-acetyl muramic acid, the network like structure will be there in the cell wall, so it is very hard to remove the peptidoglycan layer. So in gram negative bacteria, if you see, cell wall has thin peptidoglycan layer and more amount of lipopolysaccharide layer, which can be easily dissolved. So lipopolysaccharide can be easily dissolved during the decolorization process. So gram staining is based on the ability of microbes to retain the purple color of the crystal violet during decolorization with alcohol. So we are able to distinguishing them, or their gram nature depends upon their intake of the color, what color they are taking, whether they are taking crystal violet or whether they are taking the saffronin. So if you see, crystal violet iodine complex will be formed and combines to form a larger molecule in the cell wall. So what happens when you add crystal violet to the smear means crystal violet will go inside the cell wall. Then when you add the grams iodine in the next step, This grams iodine will act as a mordant, and this mordant will go and bind with the crystal violet in the peptidoglycan layer, and there chemical reaction will takes place and crystal violet iodine complex will develop, which is a very hard complex to remove. So this peptidoglycan layer will now pack up with the crystal violet iodine complex. But when you see in gram-negative bacteria, we don't observe that because of the thin peptidoglycan layer. You can see here, because of the thick layer in the gram-positive bacteria, CVI complex is not possible to remove during decolorization. But in gram-negative bacteria, peptidoglycan layer is very thin. So during decolorization, ethanol dissolves the lipopolysaccharide layer and easily removes the CVI complex from the peptidoglycan layer and makes gram-negative bacteria colorless. So all gram-negative bacteria become colorless in decolorization process. So whenever they have become decolorized, so in the last step when we add the secondary stain that is called as safranin, all the gram-negative bacteria will uptake safranin and appears red or pink in color. So here you can observe the procedure so that you can able to know what is the procedure behind. So first we need to, as like in every staining method, we have to prepare a thin bacterial smear, right? So bacterial smear is prepared on a clean glass slide, and first we need to allow to add the primary stain. The primary stain is nothing but crystal violet. So now after the addition of crystal violet, we need to keep the slide for 10 to 20 seconds because the bacteria need to uptake the stain. Now we have to give some time. Then wash off the excess stain and cover the smear with grams iodine for 60 seconds. So this is the second step. In the first step, we are adding the primary stain. We are leaving it for 20 seconds and then we are adding the grams iodine. Grams iodine addition will takes place and it leads to the formation of crystal violet iodine complex as we have discussed before, and we need to give 60 seconds. Then after that remove excess of grams iodine and again decolorize the smear with 95% ethanol. So this ethyl alcohol, 95% ethyl alcohol will act as a decolorizing agent, and we need to leave it for 10 to 20 seconds. Just 20 seconds is enough. So wash off the smear and add secondary stain, that is the last step, and allow it for 20 seconds. Let the smear air dry. So after the addition of saffron we need to allow it for 20 seconds. After 20 seconds we need to wash off the excess of stain and we need to air dry the smear. So now after air drying we need to go for the observation. Microscopic observation will helps you to observe whether your slide clean glass slide is having the smear. So that smear will be having whether gram positive bacteria or gram negative bacteria. If you see gram positive bacteria, they will appear in purple color and gram negative bacteria will observed in pink color. So like that you can able to distinguish. And what are the lens we need to use? First we need to focus under 10x lens, and then we need to increase the power of the lens to 100x. So whenever you are using 100x lens, you need to go for oil immersion. So you need to put some oil immersion and you need to examine under the microscope. So here it is a picturized representation of gram staining so that you can clearly understand what is going behind. So this is a crystal violet stain, this is grams iodine, this is decolorizing agent, that is alcohol, and this is the saffron secondary stain. So if you see here, I am adding crystal violet in the first step to the smear, right? So all the cells, they have become purple color. Then I have added grams iodine. So grams iodine has entered into the cell wall and it has turned into crystal violet iodine complex in gram positive and gram negative bacteria. And if you see in the third step, there I have added the decolorizing agent. So gram negative bacteria, due to the presence of thin peptidoglycan layer, they have became colorless here. So you don't observe color here. But all the gram positive bacteria, they remain colorful. So what is the color? The color will be crystal violet color itself. So purple color appearance you can observe. But the rest of gram negative bacteria will become colorless. Then after that I have added saffron into it. So safranin is a secondary stain. So stain, once the bacteria will uptake a stain, it won't uptake the another stain until it becomes colorless. So here these colorless cells have taken the secondary stain, whereas the first cells, that is gram positive cells, already they have uptaken the crystal violet. So that's why they don't take the safranin and they become purple in color only. And these gram negative bacteria will become red in color due to the uptake of the secondary stain. So this is a clear image where you can observe like how what happens in the each step. So here I have given some example result, like how can we expect the results after performing the gram staining. If you observe purple or violet color rod shaped bacteria, you can tell them as gram positive bacilli. If you observe pink color or red color rod shaped bacteria, you can say those are gram negative bacilli. Or else if you observe purple or violet color spherical shaped bacteria, you can say those are gram positive cocci. If you observe pink or red color spherical shaped bacteria, you can say those are gram negative cocci. So like this you can able to say depends upon their color. So whether they are gram positive or gram negative. Same time you can able to say the morphology and their arrangement also. Morphology in the sense whether they are bacilli or cocci. Arrangement means whether they are streptococci, staphylococci, streptobacilli, like that. So I have given some pictures we can see in the next slide. So see here, this is a gram staining slide where you can observe gram negative bacilli. So you can see pink color rods. So all are gram negative bacilli. And here in this slide you can observe some of the bacilli are there which are gram negative, pink color, purple color, spherical shaped cells are there, those are gram positive cocci. And then here gram positive bacilli, both gram negative bacilli, purple color rods, pink color rods, both are there. So like this we can able to observe under the microscope. When you perform the gram stain, you can observe the images like this under the microscope. This is 100x lens where you can observe clear cut shape and also clear cut color intensity of this gram positive and gram negative organisms. Like this we can able to distinguish. So this is about gram staining technique. Thank you.